Okay, think about this for a second. What if the foundational rules of genetics, the exact rock-solid principles we all had drilled into us in nursing or med school, were just proven completely wrong? And what if, right at this very moment, scientists aren't just studying life anymore, but actually building, reproducing life forms totally from scratch? If you think biology operates by a strict, unchanging set of rules, well, it's time for a massive update.
All right, let's dive into this. Welcome back to The Explainer. Today, we're breaking down 3 discoveries in synthetic biology and genetics. And I want to be clear up front. These aren't just minor textbook tweaks. These are full-blown, paradigm-shifting breakthroughs that are actively rewriting the future of clinical therapeutics as we know it. So here's our roadmap for today. We're going to cover rewriting the central dogma, the creation of synthetic life, aka spud cells, and man-made chromosomes. Finally, we'll wrap up with what this all means for the synthetic bioeconomy. Let's start with part 1, rewriting the central dogma. Think back to that one foundational, supposedly unbreakable rule about how genetic information transfers. The central dogma, right? The principle that DNA makes RNA and RNA makes protein. We have literally always been taught that to synthesize new DNA, you absolutely, unequivocally need a pre-existing nucleic acid template. Without a template, you can't build the ladder. But yeah, actually, scratch that. Not anymore. The traditional textbook rule demands that nucleic acid template, but there's a groundbreaking new report out of Stanford University that is just stunning the scientific community right now. Researchers actually discovered a bacterial defense enzyme that synthesizes DNA entirely from scratch. So instead of a nucleic acid template, it uses its own protein structure as a physical guide. Basically, it's building the DNA using itself as the blueprint. No way, right?
Now, what's really interesting about this is that it's a fundamentally new way that life produces DNA. It completely defies our basic expectations of how genetics work, just completely bypassing the traditional requirement. And sure, while this sounds like fascinating, high-level theoretical science, the real-world implications here are huge.
For those of us in the healthcare field, this isn't just academic trivia. This specific mechanism could eventually lead to next-generation bioengineering tools that realistically rival CRISPR. By harnessing this template-free DNA synthesis, we are unlocking highly advanced new ways to target and treat genetic diseases directly at the bedside. It's a real game changer. Moving right along to part 2, spud cells and synthetic life. We're shifting gears from finding convention-defying enzymes to actively building entirely new artificial cells from a soup of raw chemicals.
36. Now, that is a microscopic number when it comes to genetics, but it represents a gigantic leap for synthetic biology. To put that into perspective, a team of synthetic biologists at the University of Minnesota recently achieved what a lot of people consider the holy grail of biology. They developed these simple, human-made artificial cell membranes, and they loaded them with a highly minimalist custom code consisting of exactly 36 genes. And they playfully nicknamed these creations spud cells, mostly because of their distinct, lumpy, kind of potato-like shape under the microscope. But hey, don't let the cute nickname fool you. These spud cells represent a massive breakthrough in creating life from raw, inanimate chemicals.
Let's break down what these spud cells can actually do. While they aren't fully independent just yet, they do still need to be fed ribosomes and enzymes to survive. The rest of the process is just astonishing. Once they're fed, they consume that energy to expand and grow. And then they actually divide and reproduce for multiple generations. We are literally watching human-made cells carry out the fundamental life cycle. So why does a reproducing synthetic cell matter for your clinical career? Because we are inching so much closer to bespoke biology. Pretty soon, we'll custom-build synthetic cells from scratch to manufacture highly complex pharmaceuticals, target tumors with astonishing precision, or even act as microscopic, highly controlled drug delivery vehicles, perfectly navigating the human body. Which brings us to part 3, the man-made chromosome. This is where we start treating bacterial species less like simple organisms and a lot more like hardware waiting for an operating system. Here is the incredible process researchers are using. First, they strip down a bacterium's natural genetic code, essentially wiping the hard drive blank. Second, they replace it entirely with a custom-built, lab-synthesized chromosome. And finally, they boot it up. They are basically turning on a living, breathing computer that functions exactly according to human-written code. So, the crucial point here is how this solves massive clinical limitations at the bedside. I mean, imagine deploying synthetic bacteria engineered specifically to hunt down antibiotic-resistant superbugs, or man-made microbes perfectly monitoring gut health in real time, even synthesizing medications directly inside a patient's body, exactly when and where they are needed. And that leads us to our final section, Part 4, the synthetic bioeconomy. When we take a step back and combine template-free DNA synthesis, reproducing spud cells, and programmable chromosomes, we're looking at an entirely new landscape. Like the quote says, these discoveries are crossing the line from science fiction into clinical reality. As healthcare professionals, ignoring these advancements just isn't an option. Whether your future is in the lab, at the bedside, or analyzing healthcare market investments, the synthetic bioeconomy is for sure the next major frontier. Which leaves us with one incredibly important question to ponder. Are you ready for the next major frontier in clinical therapeutics? The rules of life are literally being rewritten right now. To keep your clinical knowledge sharp and stay ahead of these industry trends, make sure to like and subscribe to Sherringford TV and head over to sherringford.org for more career resources and market trends. Until next time.